Good afternoon, everyone. And I'm Renee, the last speaker of today. And it won't be long, so I just want to go through the main idea of my research. And I'm basically based in the Hong Kong Children's Hospital, and I just have a meeting, so that's why you can't see me in person. But I'm happy to meet you anytime if you want to talk to me in person. Or actually, one of my colleagues, Dr. Joseph Chen, is in the audience, physically present in the Prince of Wales Hospital. That he has been working with me for more than 7 years, and so he knows a lot of things within our team. So you can ask him about many things. Okay, come back to the research topics. So my team actually work on epidemiology of the respiratory viruses in hospitalized children. And more recently, we want to also look into what is the picture in the community. So this is number one. Number two, we develop cutting edge respiratory disease models. So what are these models for? I'm not talking about mathematical models, but I'm talking about cellular models. So I believe that everyone during the COVID, you have, no matter going to the station to swab by other professionals, or you do cell swapping for your rapid antigen test kit, you know about the nasal or nasopharyngeal swab. So actually we can derive cells from all these very easy obtaining sample type and then make them back as the cells that you see in our airway. Apart from differentiated cells, we can also derive some mini lungs, which looks like this in the bottom, and we call it organoid that I will illustrate later in the later slides. And the third program here running is that we do have a nasal strip little sampling tools that also mentioned by Jenny earlier on, that we also look into local immunity. So previously, usually people talk about immunity, they are talking about the circulatory immunity. While in respiratory viral infection, local immunity also takes its role. So during the COVID period, we make some tiny sampling tools and then apply it to bedside. And then we know the longitudinal changes of the local immunity that I will also talk about it in the later slide. And here we show that some of the undergraduate students during the COVID period, we also have community programs that we bring our tools to the schools and then help to characterize some of the community children's local immunity aspect. So that is generally we got three aspects. This is a slide introducing where we work and how it looks like, that I believe that Professor Leung Kam Tong just now might have also shown some of those. Apart from the Prince of Wales Hospital, which is located next to the City 1 station near Sha Tin, within our lab, we got one extra block. In the lab block, we got an extra laboratory space, which is specifically for pediatric respiratory research. And we got another satellite site, which is in the Hong Kong Children's Hospital in the old Kai Tak Airport. So this is the clinical block, while this is the research block. We are located on the 8th and 9th floor, and here a bridge that connecting us with the clinical case here. So we are at a very proximal places which we can conduct research in the lab space, which are quite actually new, and with modern technology machines equipped here in the lab, and also close proximal with the patients. And this is just one of the pictures showing you our lab space. Now we come to the first topic, which is about the epidemiology of respiratory viruses. Indeed, we are about to publish a new paper describing the phenomenon of infection after the COVID period. And now this slide shows you horizontally is the different weeks of the year across 2014 up to 2020. And the gray bar shows you the number of sample being tested in the in every single week. And you can refer this number by referring to this right-hand side y-axis. And I'm going to show you different viruses positive percentage over time. So number one, I'm going to show you the influenza virus, which is represented by black color, H1N1, a little bit gray color, H3, and then the pale gray color, which is influenza. So people are very worried about influenza before COVID. So most of the time when you hear about respiratory virus, most of the time you hear about influenza. So I would like to indicate that in Hong Kong, before COVID time, we usually have a biennial peaks of influenza. And if you see that it can cause some problems sometimes, that it goes up to 40% of positive rate in some particular seasons. The second one are the green lines, which represent the parainfluenza virus 1 to 4. And this parainfluenza virus is one of the quite prevalent virus in pediatric patients. The next one is adenovirus, which is representing in this brown color. And then the next one is also a pediatric well-known respiratory syncytial virus, which in short called RSV. And usually children get RSV at least before 5 years old for one time. So RSV is a virus that got extreme attention when you are talking about pediatric research. The next one, you see that the pink color is only appears after 2018, which is a newly included human metapneumovirus in the HA system. So we have those record after 2018, and you see when compared to other viruses is around the rate of RSV a long time. The one that is lastly shown in blue is the research interest of our team, which is called enterovirus rhinovirus group. And it's actually quite comparable with influenza, and you see a biennial peak, summer peak versus a late autumn peak. And during this very critical period, 2020 to 2022, which is the time that every one of us are wearing masks, you can see that even we got such a social distancing, this very troublesome EVRV is having its normal peak as before. And sometimes these particularly exaggerated high positive rates is because the other viruses are actually missing in the community, and they stop circulating because of the COVID-19. So we do some of these epidemiology study, we observe the patterns, and we try to pinpoint what is the potential reason, biological reason, and study the mechanism behind. And this is the database from the CHP, and we got an in-house Prince of Wales Hospital inpatient database as well. So when we are looking at viruses, what we can do, we can do multiple different ways. So we can do molecular epidemiological surveillance. If we are looking from the community, as what I showed you in the last slide, this is the one within the inpatient. So those are patients taking, having infection and then later on admitted into hospital. When we try to categorize them into different age group and break it down into different virus category, then you will see RSV stand out in the first two age group. And then eventually this EVRV actually having a high rate along the time. So however, you seldom hear about EVRV research because of its seems like not important role, because people believe that it's just a cold that it will be recovered after some time. But as what Edna suggests just now, actually there's an EVRV infection also associated with the risk of asthma development in later age, especially when you have early and repeated exposure to EVRV. And if we do the further resolution typing and try to understand the distribution of RV type in the inpatient population, RVA and RVB actually contribute half of the samples we obtained, while the other half is called rhinovirus C, which is discovered only for 15 years, but very limited studies are working on RVC because of different reasons. Number one, it is not able to be propagated as easy as we do RVA and B. It needs some special receptor that only present in the primary airway cells that I will elaborate further in the next slide. And to study all these epidemiology and also the genotyping, we are the first student of the team published the epidemiology study of enterovirus D68 within Hong Kong as the first study. And another colleagues with a postdoctoral research associate also using all this data to identify that there is a suppression of influenza virus infection by the rhinovirus interference. We do it at the population level. That means looking at all the inpatient data, and then we do individual level, and we study individual patient whether they have a higher chance or lower chance of co-infection of the two virus, and validate it at the cellular level by the models that I mentioned earlier, that we do a stepwise sequential infection in order to see what is exactly the effect. And this is Mas, who is the soul of the team that help every generation of our students in the team to acquire different skills. And as what I mentioned to you just now, we find a monopoly of the rhinovirus during the COVID period. And when we further do the classification, it is very special to find that it's a minor group virus that happens in the airway that is never observed before. So in the general year, is a multi-genotype kind of composition in the community and in the inpatient population. However, during this COVID time, then we see the minor group overrepresentation. And this is the work done by Jason, which graduated last year, and currently he is a postdoctoral fellow in the team. And as I just mentioned, if we want to study the virus pathogenicity, that is the disease mechanism and the severity of disease, we can recruit human volunteer and drop the virus into their nose, but usually it can't be done. It is done in some Europe country as a randomized control trial, but those are most likely adult samples. So it is very uneasy to have pediatric sample to study the differential immunity between adults and pediatrics. Another option, you may consider animal models, but still they are not human. Some years ago, we studied neonatal mice, which is really small. This is my thumb. So you look at your own thumb, you can imagine how small the animal is. So because of the different stage of development of the immunity, then we actually need to select a very precise model to represent the interaction between viruses and our children. So lastly, the surrogate would be using primary cells. For those who study biological science in the past, that you may remember there's something called cell line, which are immortalized cell lines that everyone is using, like HeLa cells, whatever. But in our team, we are very interested to biobank a huge range of human respiratory cells from the upper tract, that is your nasal or nasopharyngeal brushes, or from the bronchi or the lung. So when there's a surgery going on, then we already get a set of consent that we can recruit human subjects for them to donate the residual removal part of the organ. So we can start everything from the original specimen and eventually derive that into a mini trachea, mini lung, or mini nose. This is the graph showing you the age distribution of the lung tissue we collect in the past. You can see that most of the sample we have are from elderly, because most of the time when people have the lung loop removal, could be some cancer related disease that happens in older age. And recently, and when we study them, we know that it is not representing pediatric population. So later on, we start to have the upper tract sample collection in a more frequent way, and then we can successfully lower down the sample age to have a more physiological relevance with what we want to study, which is the respiratory virus in pediatric airway. What we can do with all these clinical samples, if we got 3 mL of peripheral blood, we are able to do the neutrophil study, the peripheral blood-derived monocyte, and eventually mature macrophage study. If we have lung tissue, we can do alveolar macrophage isolation, and also those distal lung epithelial cells isolation and culture. For the upper airway and the conducting airway, we are not only happy with monolayer of cells, but we derive them in a pseudo-stratified layer, which is just look like your airway that got cilia and got mucus secretion that mimics the exact interaction with viruses. To make this at a higher resolution, if we stain the cell layer with different cell markers like P63 for the basal cell markers, MUC5AC for the global cells, which are now here in red colors, and the ciliated cells now in green colors, and the club cells in green colors as well, you can see that what we culture are actually recapitulating the physiological cellular composition of our airway. So this makes us advantage to study various interaction of virus and these cells. And during the COVID time, we compare the adult and pediatric airway cells in their morphology, in the cellular population, and the receptors, virus receptor expression, and the protease and different genes related to COVID, and try to delineate the difference of responses when adults and children having a COVID infection. And we do bulk RNA sequencing, which is a method to understand what the genes expression after infection. And we compare this differential gene expression between pediatric and adult, and found that actually when both adult and pediatric cells are experiencing COVID-19, they intrinsically react differently. So these bundles of pathway are upregulated in children's, while in adults they have another set of things happening. So this can partly explain why the pediatric subjects versus adult subjects are having differential severity when they are encountering COVID-19. So after, apart from COVID-19, coming back to rhinoviruses, as I mentioned before, rhinovirus A and C. A is the classical virus that people always use to study and representing what RV do in the airway cells. While RVC is the difficult virus that we try to characterize it. So Ben is now year 4 already. So he's doing a comparison between the effect of RVA and C in the cells. So we create all these cells by recruiting true pediatric patients' cells, and then we derive them into pseudo-stratified layer. We infect them with different viruses. We do total viral RNA, total RNA extraction sequencing, and do bulk RNA sequencing. And at the same time, we do single cell RNA sequencing to understand individual clusters cells, what they are doing, and what is if any of the cluster is responsible for specific outcomes. And we found differential genes induced by RVA16 and C15, and they also got some common genes that is both virus can upregulated or downregulated. In the single cells study, we also cluster different kind of cells, and we can understand individual cell type got different responses. Such as in this figure, we have basal cells, global cells, and other cells that I showed you in the earlier biomarker study that within this epithelium, we recognize they are present. And you can still see my screen, right? And we have some image video to show the cilia movement of the cells. So before infection, you can see a very synchronized cilia movement in the culture. But after infection for 48 hours, you can see the very limited and unorganized beating of the ciliated cells. So apart from transcriptomes analysis, we also do the actually physiological measurements of the performance of cells. Those are the air-liquid interface culture I showed you. But more recently, we derived the airway organoid, which is a mini organ kind of structure that allows us to explore more things. And it got its niche that I can discuss with you because those are unpublished, that it had an advantage when compared to ALI system. So we also use this system to investigate different virus infection, including rhinovirus infection. And this video shows you how these organoids work. So after the culture of air-liquid interface, we derive them into matrix gel and they form organoid. After forming organoid, you can see the whole layer of epithelium is reconstructed and the ciliated cells are inside. So if you come to the lab, you can learn all these technology to figure out your own research question on respiratory infection. If we cut open the organoid, then we will see more things. In the interior, you see the ciliated cells markers. As what you see, they are beating inside. And if we do CSEC, you can see different cell distribution. And some people will say, hey, your airway is not in the, our airway have a lumen, while your lumen is an enclosed cavity. How can you get the virus into this physiological structure? Then we have an apical out model that all the cilia are now coming out. And when we do infection, then it's become physiological. So if we have even a higher resolution, you can see the red dot at the tips of the cilia, which is actually the very difficult RVC virus receptor. Without this receptor, you hardly get RVC to infect your cells. So we have all these models, and then we can do a good infection experiment. After talking about the cell model and the epidemiology, the last few slides, I'm going to talk about the mucosal immunity. So with the support of industry, together with the Innovation and Technology Fund, we derive a nasal strip, which is like a filter paper tools, and then inserted to our children's nose, and then we can collect a lot of information, which was previously not able to do. This allowed us to collect antibody, and because it's non-invasive, we can have longitudinal collection, follow up, and even the parents can bring all these nasal strip back home, and they can do all these measurements, sample collection, and then they can mail it back to us in the laboratory. And these tools allows temperature, room temperature stable, room temperature stability for the sample to ship from home to the lab. And it opens up a lot of longitudinal study opportunity. So with this nasal strip, we published 3-4 papers on the SARS-CoV-2 period. Number one, it's similar to nasal swab that it can detect virus. Number two, it detects different immunoglobulin type, isotype, including IgA, IgG, which is represented by green and orange color here. And we can do longitudinal study, and we can detect conjunctival and the nasal fluid, as well as the plasma antibody, and try to link the local immunity together with the systemic immunity. So Sao Jun is the very bright PhD student that is about to graduate this year, and she take all this information to do international conference and present. And every single conference she attend, she got the best poster prize. This is one of them, and this is another one of them. So her work is well recognized internationally. And last but not least, our group actually published more than 94 publications in the past. And sorry that it wasn't translated yet, but we train lots of research students, not only postgraduate, but also undergraduate students from CUHK or other university. And we take in summer students. So within the lab, you can encounter different people from different university to enrich yourself. And yes, and this is the team. So sorry about the low resolution. So currently we have 4 PhD students in the lab: Chong Li, Wang Suo, Basa, and Ben is about to graduate, and Sao Jun here is about to graduate too. So we got 2 postdoctoral research associates, and we got Kathy, the scientific officer, to assist you during your pumpy journey of being a research student. So here's my email address. In case you want to ask questions, you can either email me or contact me via any means, or ask Joseph at the PWH site. And I collaborate with many people, and here's some of those who always provide support from time to time. And that's all for my presentation. Thank you for your attention, and it's a long day for all of you. See you. Bye-bye.